All right, so we are recording and ready to go. Hey everyone, and welcome to the second biology writing workshop of the semester. I just want to note that today's workshop is going to be recorded, but no faces will show up in the online recording unless you directly speak. So if you want to chat, I'll make sure everything's anonymous otherwise. But without further ado, my name is Riley Thoen, and I'm a graduate research assistant working here depart in the Department of Plant Biology, and I'm working with the biology division here this semester to support writing in the sciences, uh, specifically writing in the biology research courses, faculty mentored undergraduate research. In the plant biology department, I'm a PhD candidate, and I study the effects of climate and patchiness on the persistence of native plant species. So this workshop series was initially designed to support writing in the biology research courses, but I know that there may be other folks there or other out there listening or other folks listening to a recording. So please note that this workshop is designed to be helpful for all undergraduate scientists at any point in their writing journey, whether they're in a research course or intro bio. And I'll just note lastly that I'm happy to help you with your writing regardless of what course you're in. So please reach out to me, rthone@uga.edu, if you'd like to do a writing conference. I'm happy to, again, discuss your writing with you, give you some feedback, any of the above at any time during this semester. So today, we're going to be focusing on writing the methods and results section of the research article. And we're going to get started here by getting this bread. I'm a huge fan of my mom's banana bread and just banana bread generally, but I would love to be able to make the banana bread my mom made in my childhood. And I want to replicate it, frankly, exactly the way she made it. But if I'm given a recipe like this, can I replicate it? Not really. Steps are out of order. There's a step missing. Step 5 is missing. We don't really know all the ingredients. We don't know the quantities. And there just aren't really details. And if you're familiar with baking, we know that baking requires a lot of good details. And that's going to impact the final result if we don't have the high level details for a recipe. A better example of a good recipe to follow would be this one. There's ingredients here and instructions to make this banana bread, just like my mom made it. And this is kind of like what your materials and methods section of your scientific paper should look like. Like someone can follow your methods exactly or your recipe exactly and replicate exactly what you did. So in other words, we want our recipe to be reproducible and rigorous, just like a method section. So reproducible in that someone can follow it exactly and do exactly what you did, and rigorous in that it does exactly what we want it to do. In this case, it'd be make good bread, or in the case of your methods section, appropriately test a hypothesis that you set out to test. Okay, so the goals of the methods section are describing how you designed your experiment and to address your research question. And you need to provide a high level of detail in your methods section so that your methods could be exactly replicated. And it's really important with that to be as transparent as possible and to use a lot of detail. So again, if someone reads your methods section, they should be able to read through it and replicate exactly what you did in the lab, and hypothetically produce the exact same results. So for achieving these optimal transparency, let's talk here about organization and style of the methods section. So first, the methods section should be written in the past tense. You already did your methods, therefore this is going to be a past tense thing. In the methods section, you can use either active or passive voice. In general, we suggest using active voice in the sciences, but we can use the passive voice here. So again, active voice is when the object is performing an action. Passive is when an action is being performed on the object. And in general, for what it's worth, I like to use the active voice in the methods section, and freely use personal pronouns. So I'll say, we gently vortex the sample, we added acid to the solution. Next, your method section should be in a narrative structure, so it shouldn't be an enumerated list. It should really be something more like a journal entry that describes what you did rather than a protocol with which is like a directive for someone to follow. This narrative structure can fit a lot more information in it, and again, it's past tense, someone's not following it directly. Like the recipe example, we don't want to do that as a list. And then lastly, I really suggest using subheadings to break up your method section. So a lot of times your experiments or my experiments have multiple different parts of them. So for instance, I do stuff in the field, I do stuff in the lab, and I do data analysis. So my method section might be broke up into field methods, lab methods, and then of course, lastly, data analysis. All right. So moving on to more of the methods section guidelines. It's really important to provide details of your experimental design. Again, like I said before, and I'm going to say it again, probably, it's important that someone can read your methods and be able to exactly replicate it. So here's some specific details you'll need to include. First, you'll need to include the materials used to conduct the study and what specific settings they are. In a lot of cases, for like really nice or intricate machines, you actually may need to cite the manufacturer that made that machine or made that material. You don't need to do it for everything if it's like, a test tube. Additionally, you need to make sure you include all your experimental conditions, things like the volume of liquid, speed of a vortexer, etc., can really affect the results of your study. And additionally, you should also note any negative and positive controls, such that someone can replicate those, and if they can replicate those, then they can compare your other results to them effectively. Additionally, you'll need to include information on your study subjects and study organisms, basically maybe where you got them, so like what population you got them from, what location, coordinates, for instance. Or also how these organisms were maintained. For instance, what were the greenhouse conditions you grew your plants in? Of course, with all the details that you're going to be including in your method section, sometimes it's not clear why you're going to be doing some of those things to an outside reader. So it's important to explain yourself and explain why you did certain methods. And this is true especially for any novel methods or methods that haven't really been reported before. If anything that has an unclear meaning or might not be someone who is a broad audience reader may not exactly understand why you did something, it's important to explain why you did. So an example of that would be, why did you cap a test tube after adding solute X to solvent Y? We added solute X to solvent Y and immediately capped the test tubes. Okay, so that's the method there, then the explanation. Capping the test tubes ensured nothing would leak because solvent Y evaporates quickly. I think this is a really great example of an explanation for your method here, because just saying like capping a test tube seems like a pretty innocuous methods thing. You wouldn't expect to need to report that, but in this case, it actually was really important so that our solvent didn't evaporate. On the other hand, if you're using an already established protocol, for instance, a protocol that is already detailed in another paper or a manual, for instance, you can cite that protocol. You don't have to go into too much detail on it. So if a reader is going to want to sort of follow your method section instead of directly following all the detail in your method section, they could go to that protocol that you just cited and find the details of that protocol there. Of course, if you make any alterations to that protocol, you will need to note that so that someone who is reading your method section would know what to do to replicate it. So for instance, maybe we can say it's an extra step. To ensure the purity of our RNA, we added a second ethanol wash step. Maybe the DNA extraction or the RNA extraction kit only had one wash step, but they did another one here because this species didn't have a pure first round of RNA wash. All right. So this is another really good example. For all samples, genomic DNA was extracted from the thorax using a standard phenylchlorome isoamyl alcohol protocol with RNase digestion, EG79. So the EG79 is the citation there. And here, they're just basically citing an established protocol, this standard phenol chloroform isoamyl alcohol protocol. But they're citing it right there, talking about it, citing it. They're adding this extra step in there, and they're also specifying this tissue type here, which is actually quite important because thorax you get a lot higher quality DNA from than any other tissue. So they're saying this here, so someone else who might replicate this study or do a similar study would know to extract DNA from the thorax with this assay. So a really good way to help explaining why and to really calibrate your readers to why you're doing certain methods is this format called to do X, we did Y format. So in other words, this format helps you clearly present your objectives and your approach. To do X, to do your objective, we did Y, we did this approach. It's really good to use these at the beginning of each paragraph because so you're letting a reader know essentially what they're going to be reading about in the next section. You may be going into a lot of detail, and this is really, really good for broad audiences especially, so like a not expert reader, as they may not understand the complex details of your methods. Notably, this is something that you can actually restate again in the results section, and we'll talk about that a bit later. So in this example here from Landris et al. 2013, the authors are basically describing how they're analyzing their data. So I start off by saying, to study the effect of background on spider color change, the objective, we used linear mixed effects model with color index as the dependent variable and background, et cetera, et cetera, et cetera. So to do X, their objective, we did Y. All these details on methods. And this is another really good example with the same type of thing. To determine the effect of molting on color change from yellow to white, we used this 80 field collected spiders that were initially yellow, And then they go into more details here below in the methods section. So again, this 2 to X we did Y format is really useful as for beginning each paragraph, really as a topic sentence. Again, calibrating your readers to why you did everything in the following paragraph. Okay. So up next, I'm going to give you all maybe a minute and a half, 2 minutes to read this methods section and find from it the following questions. First, look for objective and approach, what data they collected, and where do they provide context and justification for that data collection. All right, give you about 90 seconds here, starting now. Okay. So first, looking at their objective and approach. So here, their objective is stated in the to do X, we did Y format. Their objective is to capture molecular variation at loci involved in signaling, production, and reception with respect to the rest of the genome. And then their approach is stated just thereafter in that to do X, we did Y format. Additionally, the data they collected is actually in that approach section. We performed dd-rad-seq on individuals from these 12 populations, and they also note the sample size of that in more detail, kind of below the 2 to x weighted Y format. And then lastly, here in this final sentence, they say only male measurements and specimens were used since females are difficult to locate in the field. This is a justification for data collection. You may look at their data set and say, why are there only males here? That doesn't make any sense. But they justified why they did that in the methods, and it actually is a pretty reasonable thing for them to do, considering that females are really difficult to find in the field. Maybe they're not flashing like the males. Okay, so one last thing for the methods section before we get it all summed up is just, it can be hard, frankly, to figure out which steps in your method section you want to actually report. Because there's tons of details you do in a protocol, there's tons of details just generally in your science. And it's hard to understand what's important. So one thing you can ask yourself is, which steps would you need to know to understand how the experiment itself was conducted? So for instance, some of those steps might be implied. As an example, to collect rigorous data, you need to record your data in a data sheet or in a lab notebook. But that's implied, and we don't report that in the methods section. So what I'm going to do here is give you about another 2 minutes here to work through this example. This is essentially a lab inspired by a lab from Biology 1108, and it describes the steps a student might take when they're actually doing the experiment. Again, this is not a methods section, this is more so a protocol. So try to identify which of these steps are key steps, meaning that they directly explain the experimental design. All right, and I'll give you about 2 minutes here, starting now. Again, you're looking to identify the steps that are a key part of the experimental design to report in the methods section. All right, so here are the following steps that I identified as ones that are important to include in the methods section. So for instance, we wouldn't include steps 1 or 2 where we talk about organizing stuff on a lab bench. Again, that's something that's kind of implied in the methods section, that if you're going to be doing this experiment, you would have to organize your stuff in some capacity. Same with labeling tubes. However, to remove a clipping from the plant that was grown in soil, cut the stem with a blade that is at a 45-degree angle, and that's an unrooted clipping. So that is a really important aspect of this experiment itself. You need to do this, follow this exactly to replicate this experiment. It's important to report. So that's an example of stuff we'd want to report versus not from a protocol. Lastly, I'll note here that these two steps where we wait a half hour, then wait another hour, are kind of repetitive with one another, and then do the same thing where we record and measure. So we don't want to say that twice in the methods, and we can make it a little more concise writing-wise. So we can say instead, after initial observations, we measured the distance the dye traveled from the roots after 30 and 90 minutes. So essentially, you're combining these two repetitive things into one sentence, one statement. Okay. So as a recap for the methods, the overall goal is to describing, key steps in a narrative format, the key steps of your protocol, key steps of your methodology, again in the narrative format. So one thing that might help you write is by making a flow chart of your methods, and this can serve as kind of an outline of your methods directly. And I always recommend doing outlines. They're really helpful for me. And so a flowchart here specifically for your methods can be a really useful tool you can use. Additionally, getting feedback from other folks, whether it be in a peer review or from lab mates, can always help you identify things that you might need or might not need in your methods section. And lastly, of course, for your methods, you want to aim for rigor and reproducibility. All right. So let's move on here to the next section of our paper, the results section. So overall, in our results section, what we're doing is we're reporting our data objectively. The results of our study, the findings of our study, in an objective way, without any bias or interpretation of those results. The only thing we can kind of say in the results that's maybe a little bit of an interpretation is just saying, this result did or did not support our hypothesis or prediction. In the main text or the body of your paper, you can share your results with your reader by describing any sort of patterns and trends, and you also want to report any statistics in writing. But additionally, you're also going to generate figures and tables of your data. And you will also include these in your paper, of course, in a different section. But overall, you're going to need both the written results section and the figures.
Oh, I was like, all right.
So you're going to need both the written results section and the figures and tables. Overall, what you're doing with the results section is you're being transparent about what you found, and allowing readers to interpret your results without your bias. Again, in the discussion, you'll be really interpreting the results you found. All right. As for some guidelines for the results section, the first thing is, like the methods section, you already gathered your results. They should be written in past tense. Also, your results should be written in the same order as your methods section was. You should use subheaders that essentially are the same as in the methods section, that not mirror it, but follow it exactly. And similarly, you can use the to do X, we did Y format to sort of recalibrate the reader to the methods you did to obtain the results you did. So like I said before, in your results, you're going to be providing an in-depth summary of what basically your results show, but in writing. You really want to cue the reader to specific patterns or trends, because the readers didn't collect or analyze your data. So it's really important to just clearly communicate what you found. And additionally, like I said, you need to also make written results section and also have a separate figures and tables section. And notably, the written results section, where you're writing your results, the high-level summary of your results in text, should be able to stand alone from the figures and tables. In other words, a reader should be able to understand what you found from just the written results section and just the figures and tables. So let's take a look at this example here. Just give this a little read here and ask how the paragraph is organized. What is their approach and what are the findings? I'll give you about a minute here to read this and think about these questions. All right. So first thing they did here is they used the 2 to x, we did y format to examine variation at signaling loci in the context of the environment. We did this approach. So essentially they're recounting their methods to sort of recalibrate the reader to what they did. And then they talk more about the findings. First, they used a phrase here, this as predicted, or contrary to, for instance, is another way to say it. They found this result. So that's a thing you can do in the results section is basically recount your hypotheses and say whether or not your results support the hypothesis you made. Additionally, they start really broad on their results. So they say, as predicted, all 3 loci showed sequence variation in coding regions. And then they get into a ton more detail below on that there. Notably, they also do a good job of including the specifics of their results, like certain genes or certain exons, introns, etc., in parentheses. Base pair length, these, including things in parentheses, not in the main text like of the sentences, can really make sure your results don't get convoluted. We'll work on that in a bit. And then lastly, one thing they do is they always reference figures and tables in the text. So they say here, they say the populations differed at the peak emissions, and then they reference figure 1. So you need to make sure you reference every single figure and table in your text like that. Okay, so now we're going to pivot to sort of following this format a bit. We're going to work on basically writing results clearly in a scientific style. So my question for you is, which of these 4 following examples most clearly communicates results in a scientific style? Is it A, figure 1 shows flowers with yellow petals are more than twice as likely to receive a visit from a pollinator as blue-petaled flowers? Significant differences in pollinator visitation between flowers with yellow and blue were found. A p-value of 0.01 showed yellow flowers were more likely to receive pollinator visits than blue flowers. And lastly, pollinators visited yellow flowers at a rate twice that of blue flowers. A few seconds here to give this some thought. So, for me, the best section at communicating a general result option is option D here. I want to talk about why that is. So first, this statement focuses on the variables themselves. The variables being the response variable being pollinator visitation rate, and the treatments being blue and yellow flowers. Variables are the core of your study. They're what you're asking questions about, and it's important your results focus on them, because that's what a graph does. Additionally, this gives a quantitative comparison between treatments. And at the same time, it doesn't overcomplicate that quantitative comparison with stats directly in the sentence. So again, you have a rate twice that of, which is a quantitative comparison. You have a twice as high of a visitation rate, but we get the specifics in these parentheses, so it's not, convoluting the flow of the sentence. Next, we don't really interpret anything here. We don't say, oh, this means that, blue flowers are useless, something like of that variety. Not like anyone would say that anyways. But you're allowing the readers to read this and interpret this data for themselves before you actually talk about it in the discussion. And then lastly, this is giving a really high-level summary of your results, which is a useful way to start off presenting your results. So you can get into more details below. And what's nice about this is that it can stand alone from a graph. Someone can read it and understand exactly what you found. But also, this can pretty much be translated exactly into a graph. So these are, again, like I said, 2 standalone sections where essentially we're seeing yellow flowers had twice the visitation rate of blue flowers at this exact number. Great. And again, our variables, flower color and pollinator visitation rate, exactly in the sentence. So this can be a really effective way of basically writing your results section to match, to stand alone from your figures and potentially tables. Okay, let's get into some of the details of this. So what makes this sentence adhere to scientific style and is effectively written? So first, like we introduced a while ago, it's written in past tense. Visited is a past tense verb. Next, the subject of the sentence is the treatment, organism that you studied, or the dependent variable. So in some capacity, you're focusing on the variables studied, which again is what we report in figures anyways. Next, it includes a comparison word. So you're comparing between these two groups, in this case, two treatment groups, in this case, blue and yellow flowers. And you're also including an effect size being the relative difference in this case between groups. So that being twice the visitation rate. And we can call this a quantitative comparison statement. You've probably heard this term if you've been in 1108. But this is a really useful term for reporting results because it kind of gives a visualization to readers just in the text. And then lastly, the statistics are, and any sort of figure mention are in parentheses here. So you're not convoluting your sentence with these things. You're not having them get in the way of communicating your results. Okay, so what we're going to do now is I'm going to give you all about 2 minutes here, maybe a minute and a half, to write a results sentence like I just did in the scientific style. And just note here, there's no right or wrong answer. It's going to be up to you to check your own work. I'll give kind of an example of what to include after the fact. And in this case, I just want to note for interpretation purposes that y equals 2x essentially means that for every one additional leaf that a plant had, they had 2 additional flowers on average. All right, I'll give you again about 90 seconds to work on this sentence. Okay, so let's check on this. I'll ask you these following questions. Is your sentence written in past tense? Is the subject of your sentence treatment, organism, or dependent variable? Does your sentence include a comparison between groups or some sort of relational preposition or prepositional phrase, and an effect size? Those two being, again, the quantitative comparison statement. And does it include any sort of statistics in parentheses rather than in the main text? So again, there is no right or wrong sentence, as frankly, as long as you are communicating your science, communicating your results in a way that the reader can understand, that's great. That's all that we can ask for as readers. But this is a good little style guide for you to follow that I think is an easy way for readers to disentangle what you found in your results. So an example that I came up with from this, the sentence that I came up with, which is again not a correct answer, just an answer, is that plants produced 2 or more flowers for each additional leaf on average. Okay, so this will help us segue nicely into the next section, which is figures and tables. So in addition to the written part of your results, like I've said already, figures and tables are a really important part of your results section. And your figures and tables should be able to present your findings very clearly while standing alone from the text, the results text. Again, this is essentially what you can consider a visual representation of those results that you already presented in the body of your paper. And it's really important to make sure that your figures and tables match the type of data you have. The data that you collect must be represented appropriately according to the type of data it is, whether it's categorical or quantitative, and also match the type of science you did. I know a lot of different fields have, use microscopy, they use gels, etc. There's tons of different figures that can be made. Uh, in a lot of instances, I can help you with your figures, but it also is really useful to consult with, your TA or your lab mentor to talk about kind of the specifics of your field and how you might want to make figures to match your data type. So there's some specific formatting guides to figures and tables, and I'm going to go over them here. So all figures and tables should have a, name, whether they're a figure or a table, and a number, and the number will correspond to when they're mentioned in the text and show up in the paper. When you mention a figure or table for the first time, it's usually best to just put that figure or table like in the text close in your paper, close to where you mentioned it, or you can put it at the end of the section you mentioned it in. I'll often, you mention your figures all the time in the results section. I'll often just put all my figures at the end of the results section when I'm writing a paper. All figures have figure captions. They should have figure captions, which will go below the figure. And tables have descriptive headers. Oh, just kidding. Out of order. Yes. Tables have headers above the table. Figures have figure captions below the figure. My apologies there. And then figure captions, they begin with essentially a sentence that's almost you can consider like a title of the figure caption. It's essentially a brief summary sentence of what the figure is actually just showing. So it's describing the variables in the figure. And then lastly, it's important to mention, like we said here at the top guideline here, is that all figures and tables need to be referenced in the body of your paper. If you have a figure 1 in your paper somewhere, there needs to be a parenthetical citation to figure 1 somewhere in the written text of your paper. Okay. So here is, in my opinion, a really great example of a multi-part figure from this paper, Lindell et al. 2022, which is on the pasque flower here. It's on a study on the population persistence of the pasque flower. So notice a few things here. First, the axes of these are clearly labeled. All of the axes are labeled. I know they're a bit small for you, but it's easy to understand. Not only do they have precipitation, but they have millimeters, the measurement of it. On the y-axis, they're looking at flower number per adult plant. It's kind of a long y-axis title, but it's clearly explaining what the data in the figure is. Next, in this figure, they're showing raw data. A lot of people like when you show raw data just so that someone who's reading the paper can look at the raw data and kind of interpret it for themselves. Of course, you'll also want to include your statistical results, in this case, a regression line in their figures, but also seeing the raw data can help, a reader interpret your results in reference to whether or not maybe the statistical test you used was appropriate. Additionally, they include headers here above each figure just to specify, which population it is in this case, or to specify which each pane of the figure means in general. Real quick, two things I'll note here is one, just note they use a scatter plot here. This is quantitative data, and so they wanted to use a scatter plot to show quantitative data. So again, just make sure your graph type is matching the data type. And second, I'll note that in this case, this is formatted for a scientific journal, and it was formatted by the journal. Usually the figure caption will be below the figure. In this case, it's to the left, but note that in your papers, you should include figure captions just below the figure. Okay, so here's the formula we generally like to use for the figure caption. So like I said before, that first sentence of the figure caption should be kind of like a title almost. It's a concise one-sentence description of what information is displayed in the figure. And in this case, in this figure caption that we just looked at, it's here. Relationship between flower number in adult plants in a given year and precipitation in the previous year in 3 investigated populations of this species over this time period. So it's, clearly saying what data is displayed and when and how that data was collected. Next, it's always important if you have statistical results displayed in a figure to include those results in the figure caption. And they did that exact thing here with their results from their 4 regression lines. I'm not going to read those, but they're there. Then additionally in the figure caption, it's really important to talk about and kind of decode any sort of labels in the figure, symbols, any derivations of data you did, so that someone can understand how you got from a data set to this figure and what everything in the figure actually means. So in this case here, they talk about how the relationships are based on yearly averages of flower number. It's important they note yearly averages because they could have also gotten this figure with just 1 or 2 individuals. Instead, they're using averages from the whole population. I'll note that in some fields, often figure captions include a kind of a brief description of the pattern in the data or a brief interpretation of the data, which can help direct the reader's attention. This isn't always the case. Some fields do this. My field, just like ecology, we don't generally tend to do this, but you can do this if you prefer. It's up to you. So in some cases, you can describe trends in the data if you'd like to. Then lastly, I'll just note the goal of the figure caption is basically such that someone can look at the figure caption and get the gist of what was done in the experiment so they can interpret the data in the figure. Again, it should be standing alone from the main body of the text. Okay. So I'll note a couple more things about figures here. So figures can take, frankly, tons of different forms. Sometimes figures are illustrations of sampling areas or a map, or they can be microscopy photos. In this case, I think it's really neat the author actually pointed to specific areas in the microscopy that you're supposed to be looking for, supposed to be focusing on. It's a really useful tactic in a figure. And I'll note too that sometimes it can be hard to actually, when you're reporting results in text, I've said kind of throughout, it's important to make sure you're reporting results in text and in figure form, and you should always do this. But sometimes, in the case of this map and in the case of this microscopy, the figures are going to be doing a lot of the heavy lifting. Like you should still report your stuff in text, but it's often just way easier for someone to look at it. So for instance, you maybe include like a table with the coordinates of this or something in the text, and then but actually looking at the map is a lot more useful. And like I said, just make sure you're knowing how the different data types you have dictates what type of plot you'll make. Again, I suggest talking with your lab TAs or your mentors in your labs to help you with this. But if you have any questions, I'm also happy to consult you on making figures and doing data analysis for your projects. Okay. Now, in addition to tables, scientific papers often also or figures that we often include tables in our scientific papers. And usually we use tables to report the results of large statistical tests or any sort of descriptive statistics. In this table, they actually even put little figures in, which I think is awesome. And tables in general, they can have kind of similar-ish axes to figures with columns and as like, various statistics you want to report and rows as different sort of treatments or groups. And you can switch those around depending on what's most convenient for your table. I will note, though, that if you're going to use a table, please make sure that your tables are not conveying identical information to your figures. Because figures are usually going to be a lot easier for a reader to visually interpret than a table. So if you're reporting, for instance, the mean across groups in a figure, making a bar plot, you probably wouldn't want to report the means in a table of those same groups. It's kind of repetitive and figure is going to be easier to interpret. Okay, so we're about wrapping up here, but I do want to mention a few things before I go. So first, if anyone's interested, they will be doing an optional peer review for the biology research courses here in the coming days. So basically, if you're interested, you can submit your methods and results sections to the Dropbox on ELC of the biology faculty mentored research course. Essentially, if you submit it, you'll be given another paper to peer review, and another person will peer review your methods and results sections. And we'll be using a peer review guide so that will be kind of focused review. Just note too, if you're interested in a peer review, but you aren't done with stuff, you can still submit your peer review without everything being done. This peer review draft, and again, the draft is due next Wednesday, a week from today, October 9th at ELC. And I do actually want to circle back here. I know a lot of people might not have their results yet. So if you just have like a methods and like preliminary results, submitting that could be really useful because you could learn a lot about what is best communicated in your writing. And I will also note, if you're not in the 4900R class, which is faculty mentored research, you can also just email me a draft of something, and I'd be happy to review it myself, or I could submit it for peer review as well. And just note that after the submission, I'll coordinate peer reviews via email. So two last things here, quick. I do want to say thanks to you all for coming, and then thanks to all these folks with the Writing Intensive program and the Division of Biological Sciences who have made this possible. And real quick, if you all would be so kind, I have basically a survey. It's a Google survey, it's anonymous, from this workshop that essentially just asks what was helpful from this workshop, what was not so helpful, is there anything you would change moving forward in future workshops? And I'll put the link to it in the chat here. And I would really appreciate if you all took the time, took the time to fill it out if you have a chance. I'll probably also email it out to folks, but I would really appreciate it if you did this. After the first workshop, I got some good feedback and have improved it to some degree, I think. So hopefully we'll have more going forward. This feedback form shouldn't take more than 5 minutes. All right. So now's the time if you have any questions to please ask. And also note these other writing resources besides myself as the TA for writing and biology. We have the Writing Center in Park Hall and the Science Library, which can really help you do background research for your writing. All right. Thanks, everyone, and please let me know if you have any questions.
Thank you, Riley.